Welcome back, and we're going to start talking about DiseaseMap. I think understanding human health and disease is a big reason that we're here. So we've got a pre-recorded talk from Anna Gloyn, who's in San Francisco. We hope that she will emerge to join a Q&A after the fact, but I am sympathetic to the hour of the morning that it is right now over there. So let's hear from Anna on the pre-recorded video.
Very good morning to you all from California, where it's currently 1:30 a.m. I'm delighted to have the opportunity to join you all virtually, although disappointed that I'm not there in person. And I'm one of several speakers that you'll be hearing from this morning who will be providing a case for why their disease of interest should be a flagship disease and setting the way for a pitch that is a flagship disease proposal where we'll all come together to work under a similar framework with under ICDA. So my task this morning is to tell you about type 2 diabetes and efforts to move from a genetic association to a molecular mechanism. And I will be doing this through my own personal lens, really drawing on my own experience and research, but also I hope to share with you the framework that I'm now part of under the Accelerated Medicines Partnership for Common Metabolic Disease. So if we look at the International Diabetes Federation Atlas from last year, they presented that 537 million adults are currently living with diabetes worldwide, and around 90% of those will have type 2 diabetes. That's 1 in 10 adults. Last year, diabetes caused 6.7 million deaths. That's 1 death every 5 seconds. 4 in 5 people with diabetes live in low-income and middle-income countries. If you look over here on the right-hand side of my slide at this atlas of the world, different regions are depicted in different colors, and at the top and the bottom you can see the predicted increase in diabetes between now and 2045. Across the world, we're expecting an increase in 46%. And if you look in different regions, you'll see that there are some really alarming numbers. For example, in South and Central America, a 50% increase in diabetes prevalence. In Southeast Asia, a 68% increase. And in Africa, a 134% increase in prevalence. So very alarming numbers, and very clear that we need to be doing a better job at preventing and treating diabetes and the complications that arise from having elevated blood glucose levels. So blood glucose regulation is very complex, and you become somebody with type 2 diabetes on account of the genes that you've inherited from your parents and the way that they interact with the environment that you live in. So it won't have escaped you that we now live in a society where sedentary lifestyles are on the increase, and there is a global problem with obesity. And that combination of obesity, sedentary lifestyle, and a genetic predisposition can set you on a path to having elevated blood glucose levels that can lead to some very nasty diabetic complications that include blindness and amputation. Your blood glucose is actually controlled by a number of different organs within the body, not just the pancreatic, beta cells and islets that are responsible for the hormones that we think of as lowering and increasing your blood glucose levels, insulin and glucagon, but we also have metabolic tissues that are important for glucose disposal, such as the liver. We store excess glucose as fat in our adipocytes, and we burn glucose in our muscles. The brain, of course, is central for controlling our appetite and satiety, and defects in these tissues, it won't surprise you to know, can be part of the mechanism for being someone who develops type 2 diabetes. So in terms of our genetic understanding of type 2 diabetes, the field has been very successful. We currently know about 50% of the genetic risk for type 2 diabetes. We have way over 600 loci. Most studies have so far been being conducted in European populations, with the most recent studies making some headway at being more inclusive of other ancestries. But type 2 diabetes is really a poster child for complex traits. Because diabetes is relatively common in the population, it's quite straightforward to assemble large cohorts that can be genotyped to allow us to do very large GWAS studies. And we also, though, can benefit from our ability to do lookups for other GWASs in terms of physiological relevant traits. Things like lipid levels, body mass index, proinsulin levels. And all of these things allow us to garner more information about the underlying pathophysiology. We've also got a pretty good handle on the tissues that we know are involved in governing glucose regulation. So this is a huge head start in terms of our efforts to assemble large multi-omic data sets. But like everybody in the complex genetic space, we're struggling with the fact that most of our signals are in the non-coding region of the genome, and we need to link that signal to a regulatory element and to a change in gene expression. But this, of course, is really complicated because gene regulation is context specific. So all of these questions need to be asked in the context of which cell type in the body, when is this happening, and does it need to happen under a particular stimulus? So for us, one of the most important tissues that we need to work with is the pancreatic islet. And we have very good evidence to believe that a large proportion of the signals that we've identified are working through this critical tissue. But pancreatic islets are not an easily accessible tissue. They're not, for example, in GTEx, and there's very good reason for this, because they are a very small proportion of the human pancreas itself, about 1%, and you need to isolate the islets from the pancreatic organ, and that's not a trivial undertaking. It largely happens when people are trying to isolate those islets to transplant them for patients who have type 1 diabetes. And on the back of that, we've been able to access islets for research. Whilst in Oxford, I was able to collaborate with colleagues who were running the islet transplantation programme. And I've also got a long-standing collaboration with Patrick MacDonald at the University of Alberta in Edmonton, who runs an islet isolation programme for research. Since moving to North America, I've been able to become part of the Human Islet Research Network and part of the Human Pancreas Atlas project, which is characterizing functionally and omically the islets from people with and without different forms of diabetes. I'm also part of the Integrated Islet Distribution Program that provides islets for research to individuals in the US and beyond. And we have a centralized platform for phenotyping and genomically characterizing those islets. So initially, most of our work was done with bulk tissue, but more recently, we've stepped up our efforts to provide single cell resolution for both our functional assays and for our omic assays, which cover genotype, methylation, chromatin state, and transcript. So back in 2015, we were able to publish our first EQTL study in human islets. This was on 118 donors. And I should highlight that we did not do any formal colocalization analysis at the time. Rather, we looked at overlapping EQTL and GWAS signals for type 2 diabetes. And to ensure some level of stringency, we restricted our analysis to those signals that were overlapping with an islet stretch enhancer. In doing this, we identified 21 E genes that included the transcription factor Z-MYZ and adenylate cyclase 5. It's taken us 7 years, though, to move from that transcription factor discovery in the pancreatic beta cell to demonstrate that it's important for glucose homeostasis. We've managed to generate a beta cell specific knockout of this transcription factor and show that it is glucose intolerant. And then we've been able to go back to our ever growing set of donors and look at the performance of islets taken from people with and without the type 2 diabetes risk alleles, and show that if you have a type 2 diabetes risk allele in Z-MYZ, you have a worse response to glucose in terms of the amount of insulin you can secrete, and you also have reduced insulin content. Recognizing that sample size matters and that it would be incredibly valuable to bring these very important data sets together, we collaborated with Manolis Dermatakis, with Steve Parker and Michael Stitzel and Leif Group to bring together our respective cohorts to allow us to reanalyze them through a single pipeline and to do a formal co-localization analysis on 402 islet donors. And with this larger sample size, we were able to identify a total of 46 GWAS loci with 48 signals that co-localized by one or at least one of two methods with an EQTL. And this included for the first time TCF7L2, which of course is one of the most important signals for type 2 diabetes. It's one of the first that we described, and it has the largest effect. What was really interesting, though, and I think is still an important point to make, is that when we look at the enrichment of effect sizes in islets versus GTEx tissues, including pancreas, which of course contains a very small number of islets, we were able to see that we saw a much bigger fold enrichment for glycemic traits in type 2 diabetes, particularly those that we know are working through the beta cell based on human physiology in islets compared to other tissues. So like many, one of the exciting things that we've been able to do is to start to assemble different types of omic data. And we've tended to do this at a locus by locus basis rather than doing this genome wide. I think that's something that we need to consider doing more systematically. But here's a nice example of the ADSI5 locus. You may recall this was one of the first signals that we found an EQTL for. And we've subsequently been able to show evidence of both chromatin and methylation allelic imbalance at this locus, and to use chromatin capture techniques with capture-C to show looping between the promoter and the enhancer where the SNPs are residing, which have been fine mapped down to a single SNP. So a really nice way that we can link a SNP to a regulatory element and show effects on gene expression through multiple different readouts. But why have we only identified 46 EQTLs? Admittedly, we know not everything is working through the islet, but perhaps there's more to find. And one of the things that we've been very conscious about is the fact that we should think about development. Now, we know from monogenic forms of diabetes that pancreatic beta cell development is very important. Many of the key transcription factors that are involved in monogenic diabetes have a role to play during development. So we've been fortunate to collaborate with Julie Snedden and her postdoc, Sean Delallo, at UCSF. And what Julie's been able to assemble is a very valuable data set on pressure samples from human fetal pancreas at 12 weeks of development. And using single cell techniques, they've been able to put together an omic map of gene expression and chromatin state in the developing pancreas cell types. We in my group with Han Sun and Seth Sharp have been interested in asking whether or not there's any evidence of enrichment of type 2 diabetes GWAS signals in the open chromatin from fetal alpha, beta, and delta cells and the endocrine progenitors. So when Han looked, he was able to show that there were over 150 differentially accessible peaks of chromatin between fetal and adult islets. There is clear evidence of an enrichment of hits in fetal islets. And when we look specifically at our credible set analysis from the latest GWAS, we can find evidence that 28 GWAS loci variants are enriched in these differentially accessible chromatin peaks. And looking down this list, it's no surprise to see that many of these are important transcription factors involved in beta cell development. So this is now a really active area of follow-up between my lab and Julie's lab at UCSF. But a change in gene expression does not mean it causes diabetes. And despite the fact we've assembled very good evidence with our cis-EQTL studies, we have co-accessibility studies, and we have now proximity assays, just because we can demonstrate a change of expression in the islet, it does not necessarily mean that this is on the causal pathway to diabetes. We know that there's extensive pleiotropy. So it's going to be very important to find other ways to be absolutely confident that that change in gene expression is responsible for diabetes risk. One way that we've been able to do this with a high degree of confidence is by studying coding variant signals. Because we've had such confidence that we're working with the right effector transcript, we've been able to dive deep into mechanistic studies. And these mechanistic studies can take us many years and can involve multiple students and postdocs and be real Herculean efforts. So it's going to be very important to have a high degree of confidence as we move from these regulatory signals to proteins that we're working on the correct one. So how can we do this more efficiently when we don't have a coding variant signal to give us that confidence in investing years of our experimental lab lives in studying a particular protein? Well, one way that you can achieve this is by scaling up and doing simultaneous gene perturbation with disease-relevant cell types and disease-relevant assays. So you might think about doing this either targeted or genome-wide. You'll need to identify a disease-relevant and scalable cellular phenotype that you can measure. And you'll need to be confident about the cellular model that you choose to do this in. It needs to be relevant for your disease. Again, you need to be able to work at scale, and it needs to be genetically modifiable. So we did a first study in this space some years ago now, which at the time seemed incredibly ambitious. We took 300 genes at 75 GWAS loci for diabetes or glycemic traits, and we decided we would look at physiological and pharmacological measures of insulin secretion and cell count. And we chose to do this in a human beta cell line that had only recently been described, but one that we had studied and shown matched up very nicely to primary beta cells. And we felt it was really important, knowing that there were some fundamental differences in the secretory machinery between rodents and humans, that we worked with a human beta cell model. So Cern Thompson, a PhD student in the lab with myself and Mark McCarthy at the time, set up miniaturizing an assay to measure insulin secretion and selecting genes within 1 megabase of a GWAS signal. He measured those 5 cellular phenotypes, and the summary slide here really is that he found 67 hits, 15 for cell count and 52 for one of those 4 insulin secretory phenotypes. This meant that we had 45 genes at 37 GWAS loci where we had evidence that they were involved in beta cell function. An important sanity check was that we noticed that there was enrichment of genes involved in monogenic forms of diabetes. And we were also really reassured to see that that transcription factor Z-Ms that we picked up in our EQTL studies was also a hit for both insulin secretory phenotypes and cell proliferation in our screen. So 300 genes at 75 loci, pretty good, but can we do better? Let's face it, we keep finding more loci, so how do we keep keeping this a contemporary list that is useful in decision-making of where to place your laboratory dollars? Well, introducing Antia Rotner, a previous PhD student, now a postdoc at AstraZeneca. And during her PhD in my lab in Oxford, she set about to perform a genome-wide pooled CRISPR loss-of-function screen for insulin content, finding out which genes in the human genome are important for how much insulin protein you make within your pancreatic beta cell. We chose to use the Toronto knockout library and to use our human beta cell line that we'd used in our previous screen. Antia infected the beta cells such that they had a single guide per beta cell, and then we grew the cells and then sorted them on the basis of how much insulin protein they had using fluorescence activated cell sorting. We could then isolate the DNA from the population of cells with very little insulin and the population of cells with lots of insulin, and then we could map back the guide RNAs to work out which genes were important for insulin content. Not a trivial undertaking. Working with human cell lines comes at a cost. It's more expensive, it's more time consuming, and it's more difficult. But it gives you a human cell line that you know you can directly extrapolate from because we're working with human data. So for 4 months, Antia grew up the cells so that we had at the end the 700 million cells that were required for 2 rounds of our screen. And at the end of it, I'm delighted to say it was successful, and using very stringent analysis, Antia was able to identify 580 genes that we know influence the amount of insulin hormone that you have within a pancreatic beta cell. So how could we use this data to give us confidence about genes to spend time on in our lab in terms of understanding molecular mechanisms for type 2 diabetes? Well, we wondered if we could use it as part of an approach to identify effector transcripts at type 2 diabetes GWAS loci. Our colleagues, Mark McCarthy and Anuba Mahajan, Elisa Manning and Brent Richards, had all come up with different methods using published data, using genomic annotation to prioritize likely effector transcripts at GWAS loci. And in the type 2 diabetes knowledge portal, these had been depicted in various ways. At the time I was first interested in this, the 4 of them had deposited their data, and I could look at a curated approach for all of their methods. The higher the score here, the more confidence that we have the correct effector transcript, and across the bottom you have the chromosome that effector transcript lies on. So we intersected this list of 336 predicted type 2 diabetes genes with the 580 from our genome screen, and we identified an intersection of 20 that were overlapping. So I was interested in working out whether or not our data would elevate our confidence for any of these genes being causal for type 2 diabetes. Now, if we look at this list, these genes that overlap, there were some that we were already really confident about. This is a glucose transporter, important for getting glucose into the beta cell to metabolize and trigger insulin secretion. But there were other genes that we just never had considered in the concept of or context of diabetes, such as calcoco2, which had been reported mainly in terms of mitophagy. So recognizing that we'd got strong data implicating this gene in beta cell function through a phenotype of insulin content, knowing that there was also data coming from other methods that prioritize this signal, we embarked on detailed mechanistic studies within our lab. We were able to show that when you lose calcoco2, you end up with fewer insulin granules, and the granules that are around seem to be mature. We have much fewer of these immature granules. We also noted that there was evidence for these large vacuoles and vacuolated structures within the beta cells that were really reminiscent of autophagy. And that gave us the confidence to test formally for autophagy. And you can see here on the right-hand side of my slide that we were able to show that when we inhibited fusion of the autophagosome with the lysosome, we saw an accumulation of LC3, which is a marker for autophagy in pancreatic beta cells. So we know from our studies now that calcoco2 is an important gene for granule homeostasis and for recycling of granules within the beta cell. And this is now an active area of research within my laboratory. So we've taken our data, not just from this genome-wide screen, but also the previously published data from our arrayed screens, and we've made it available in the Accelerated Medicines Partnership Common Metabolic Disease Genome Atlas. And this is led by Carl Galton at UCSD. And we've been working with Carl to find different ways that we can display and make our data visible to colleagues so that it can be used for their own efforts. And I think this will be a growing area of activity as we generate more different types of data from those that we're more accustomed to with omic and genetic data, and where we have these perturbation screens and we need to display and make it available to colleagues. And that really segues nicely into where I want to finish up my presentation, which is really about telling you about the infrastructure we already have in place for type 2 diabetes that has been assembled around the Accelerated Medicines Partnership. This is in its second funding cycle. The first cycle was very successful and brought together multiple groups in the US and in the UK to share genetic data and to start to build up omic resources. And in this second effort, it's really about trying to go from variant to effector transcript. So we've assembled ourselves and our teams into a number of working groups that are tackling important efforts. Firstly, integration of genetic association studies. We've got these two large studies recently, the Million Veterans Project and the Diamante Project. How do we bring that through to a single common set of signals? We have our effector transcript working group that's really wrangling complex omic data across multiple tissues. So we have a models and assays group that is really setting about agreeing standards for assays that can be deployed for understanding metabolic pathways, looking at how we can carefully select the model systems that we're deploying and making sure that we have agreement on that across experts in both academia and pharma. And then, of course, we're working with the portal team to find innovative ways of sharing these data through the portal that will accelerate our field. We have opportunities through AMP to bring other people into the consortium through opportunity pool funding. This funding can be used to identify gaps, whether that be tissues where we have got a paucity of data, whether it can be shared tools that would help accelerate our common goals across the consortium. We also have funding calls that are led through FNIH and are mainly decided by our pharma partners, and a lot of efforts going on in this space at the moment to generate omic data sets on clinical cohorts with longitudinal data. So I'd like to finish up by just focusing on one example of a project that is coming out of this effort and really illustrate how we're trying to work together and share data to perform the optimal experiment that we can. So one of the things that I'm working very closely with Melina Klausnitzer and her team on is putting together a prioritized set of signals that are important in multiple tissues so that we can design a library for a perturbation assay that can be deployed across multiple cell types. Clearly, we want to focus on signals which have got action in as many cell types as possible so that this resource is useful for multiple teams. So we're using a variety of different approaches to put together an informative list. We're using something called a tissue of action score that was developed by Jason Torres in Mark McCarthy's group at Oxford. We're using unpublished data from across the consortium that's been generated in multiple diabetes relevant cell types and tissues, looking at co-localization of eQTLs and chromatin QTLs in adipose, hepatocyte, islet, and muscle. This has been led by Karen Mulkey and Steve Parker. And we're using physiological clustering that's been developed by Miriam Adler and Jose Flores so that we can again get insights into the underlying physiological processes that might be altered by these genetic associations, because we can use that to corroborate evidence from the eQTLs and the tissue of action scores for the cell types that are likely to be at play. So we are designing a CRISPR-I library, an inducible CRISPR library that can be deployed across multiple cell types. Initially within this call, we'll be focusing on our 4 cell types that we're heavily interested and invested in. That's islets, liver, adipocytes, and myocytes. And then we'll be running them with assays that we've developed and validated within our team. But we'll be able to extend use of this library to other investigators who are working in other disease-relevant cell types. For instance, we're working with Ines Cebola at Imperial College London, who is particularly interested in liver sinusoidal endothelial cells, which she's identified some key signatures in her omic data underlying non-alcoholic fatty liver disease. So I think this shows how we can start to build this resource, bring all the data together and be able to benefit from lookups in other data sets that will improve the conclusions that we can draw from our own data. And of course, with a library that can be extended, there's always opportunities as new assays are developed to redeploy this to increase the complexity of the cellular phenotypes that we're capturing. But what are some of the gaps and the challenges that we face? And I've underlined some because I don't see this as an exhaustive list. I think that one of the things that we've done very poorly so far is our capturing of non-European populations, both for genetic discovery, but I think critically for characterization of tissues for omic analysis. Although we're doing a better job at extending our omic studies to beyond the beta cell and adipocyte, there are still tissues that are poorly represented that have got extreme value for diabetes. We've got a lot of bulk tissue omic data, and we're moving towards more single cell analysis, but there's always room for improvement there. I think we're beginning to realize that the clarity and increased granularity that we get from single cell data is worth the investment. We still have instances of data sets that are in either males or females and not in both. We need to do better there. And much of the data we have is a snapshot. We have very few longitudinal cohorts. Something that I feel is particularly lacking, and I would really like to see improved for the diabetes relevant tissues, is the ability to collect multiple tissues, pancreas, fat, muscle and liver from the same individual. But that is extremely challenging. And I think from the slide I showed you at the beginning where our islets come from, you can start to see why that might be the case. As we move towards mechanistic studies, I think we have decisions to take around whether or not we should be going genome-wide or targeted. I think we have to do better at sharing lines and libraries. We need to have conversations around knockdown versus knockout. And we need to think carefully about how we layer bespoke and generic phenotypes on the same cell lines so that we can do things in a very cost-effective manner. And of course, we need to be more ambitious and move beyond a single cell and start to think about looking at things in organoids. So some of the areas where I think that ICDA could be particularly helpful are those that I've highlighted here in bold. So how can ICDA help move the needle? Well, I think there are real opportunities, particularly with CellMap, as we've already heard, for infrastructure for shared cell models from diverse ancestries. I think in the diabetes field, we could really benefit from having common cell models and access to the generic data that's already been generated on them that we can add to with using differentiation into additional disease relevant cell types that we could phenotype with our assays that we've developed. I think ICDA could also help with the infrastructure to collate and share that phenotypic data so that it goes beyond type 2 diabetes. I think there are also opportunities for greater connectivity to diverse biobanks so that we can increase the representation of ancestries across our data, and also the ability to generate with our collaborators omic data on diverse ancestries. In terms of the disease map, as you'll hear later from John, I think there are real opportunities to have shared models, to increase data sharing and increase resources that will help us look with fresh eyes at loci that are important and pathways that are important across many complex traits. That I've presented today is by no means the work of just those that were acknowledged on the slides. It goes far beyond that. Some of the people are depicted on this slide, colleagues from the groups at Michigan, Boston and Oxford, colleagues at a meeting some years back now where we met with colleagues at NIDDK to think about the challenges moving forward as we went from association signal to mechanistic insight. colleagues in the islet cell community, colleagues at a recent meeting working in different tissues, trying to bring kidney into the discussions on type 2 diabetes with a greater granularity. And of course, hanging out with people socially is a great way of making connections. So I thank the funders and I thank you all, and I look forward to the panel discussion later this morning.
Lovely. So Anna will be joining us for the panel discussion. It's a bit of an unfriendly time slot right now, so it's my great pleasure to introduce Dr. Carl Anderson, who obtained his PhD at the University of Edinburgh under the supervision of Peter Vischer, spending the latter years at the University of Brisbane. He then moved to Oxford to do a postdoc in which he became part of the inflammatory bowel disease effort of the Wellcome Trust Case Control Consortium. Carl's a senior group leader in the human genetics program at the Sanger, where his lab carries out genetic and genomic screens to identify the genes, pathways, and cell types driving immune-mediated disease. The translation aims of the research are to identify candidate drug targets for disease and to deliver on the goals of personalized medicine. Carl, the floor is yours.
So I'd like to start by thanking the organizers for inviting me to speak today. And I'm here to talk about our efforts over 15 years or more trying to identify genetic loci associated with inflammatory bowel disease with the view of understanding the biological mechanisms underpinning the disease so that we can identify better drug targets to treat the disease and also use personalized medicine approaches to decide which drugs should be given to which patients at what time to have the most maximum effect on their disease symptoms. So I'm going to start with a very quick introduction to inflammatory bowel disease. There are basically two common types of inflammatory bowel disease: ulcerative colitis and Crohn's disease. They differ from each other in the nature, extent, and location of the inflammation. So in ulcerative colitis, the inflammation is restricted to the innermost layer of the epithelia, and it typically extends upwards from the rectum in these continuous patches of inflammation. In Crohn's disease, you get much deeper ulceration of the epithelial layers and the deeper layers of the gut. It can occur anywhere, actually, in the gastrointestinal tract, and it occurs in these patches, these discontinuous patches of inflammation in both the small and the large intestine. So together, these two common forms of IBD affect around 1% of the global population. Particularly in populations in Europe, North America, Australia, the prevalence is around 1%. In other parts of the world, actually, the incidence of IBD is on the rise, and there are certain parts of China, Central America, South America that 20, 30 years ago wouldn't have had any IBD patients, and now they're actually they're inundated with a rising prevalence of IBD within these populations. And I think that just really speaks to the need for something like the International Common Disease Alliance, and indeed the International IBD Genetics Consortium, of which I am a member, to facilitate ascertaining samples, DNA samples, yes, but also tissue samples from these parts of the world that are just now beginning to really experience IBD for the first time. Okay. So I'm going to talk to you about a few studies that we've been undertaking predominantly over the last 5 or 6 years as we try to move from maps to mechanisms to medicines. And I'm going to start by just introducing a study that we published earlier this year where we used whole exome sequencing of about 30,000 Crohn's disease cases and 80,000 population controls. And this was carried out as part of the International IBD Genetics Consortium and in very close collaboration with Mark Daly and colleagues at FIM and the Broad. So what I'm showing you here is a bunch of coding variants which, coming into this study, we already knew to be associated with Crohn's disease. On the x-axis, I'm showing you the allele frequency in NOMAD of those various coding mutations, and on the y-axis, I'm showing you the effect size, the odds ratio for Crohn's disease. Here, I'm going to show you 5 new coding mutations that we've associated with Crohn's disease risks. Now, these variants were in regions of the genome that we hadn't previously associated with Crohn's disease risk via genome-wide association studies. So this whole exome study really implicates these genes in the biology of Crohn's disease for the first time. And having actually spent the last 10, 15 years doing genome-wide association studies, identifying loci rather than genes, and then worrying about what is the effector gene in the region, how do we extract biological insights from our genetic studies, the immediate interpretability of these coding mutations was actually a real joy to behold. In green here, I'm showing you 5 further missense mutations, coding mutations in 5 additional genes that do actually lie and reside within genome-wide associated loci that we'd previously identified for Crohn's disease. And these variants are independent of the GWAS hits, but actually they very neatly kind of give us a good insight into the likely effector gene at that GWAS locus. And so if we think then across what we've learned then from this study, so PDLM5 as well as the mutations in CCR7, CCL19, and that pathway really highlights the emerging role of mesenchymal cells in intestinal inflammation. And I just want you to remember about mesenchymal cells because we'll touch on them a little bit later on as we start thinking about our single-cell RNA sequencing studies. That was kind of phase 1 of the study. We are currently working on an expanded whole exome sequencing analysis, which is going to have around 90,000 IBD cases. So this is us bringing ulcerative colitis exomes and patients into this analysis for the first time, and over 500,000 controls. And so we hope to be publishing that paper later on next year. Of course, the workhorse for human genetics over the last 15 years hasn't been whole exome sequencing studies. It's been genome-wide association studies. I'm just showing you here a little snapshot of where we are from our GWAS studies in IBD. For anyone who is counting loci, we have 241 loci that are currently associated with IBD risk. And in our latest study, genome-wide association study, which is being conducted via the International IBD Genetics Consortium, we've corralled together, well, in this kind of phase 1, which I'm presenting the results from here, 73,000 cases and over 1 million controls. And you can see from that kind of phase 1 preliminary analysis of that revised GWAS, I've identified an additional 174 new association signals. In phase 2, which we're currently working on at the moment, there's 115,000 cases and it's the same number of controls. And so I'm predicting, and it's a bold business predicting, but I think once this analysis is finished, we'll have more than doubled the number of IBD loci. So if we thought there were difficulties before, moving from genetic associations to mechanisms and biological understanding, we've more than doubled the problem, I think, by giving ourselves more loci to work through. So this revised GWAS is the first that we've really included in large numbers individuals from non-European populations. So we have about 15,000, 20,000 cases from East Asia. We absolutely need more global diversity in our genetic association studies, and I'm hoping that via the ICDA we can foster that and really improve the representation, the global representation of individuals in our studies, particularly in these communities and regions of the world where the disease is on the rise. Okay, so I just want to stop here and point out, because you might be thinking, as we say, maps to mechanisms to medicines, that the kind of genetic mapping side of all this is a solved problem. And I think in terms of the approaches, it is a solved problem, but actually so much of what we've done so far has been around disease susceptibility. It's been comparing cases to controls. And actually, we know very, very little across the vast majority of common complex diseases about disease subphenotypes, disease progression, progression and drug response. And I think we need to start paying greater attention to this. So certainly in IBD, once a patient is diagnosed, there are a multitude of different trajectories that patient can go on and can experience. So what I'm showing you here is just a little schematic where you can see in red there is a patient who has very chronic, relapsing symptoms where their disease activity very much kind of oscillates up and down quite dramatically. In yellow, I'm showing a patient who has a relatively quiet, kind of quiescent disease course, then slowly gets worse and worse over time. And then in blue, a patient who basically just has chronic, continuous symptoms. And we do not understand anything about the genetic regions that are associated with this variability in progression. And so I think this should be something which, across many different common and complex diseases, we start paying increasing attention to. And within the UK, we have a cohort of 35,000 IBD patients within the IBD bioresource. And we have very detailed clinical and demographic data on those patients. They're consented for recall by genotype and by phenotype. And we also have funded via the NIHR the Gut Reactions Data Research Hub, which allows us to collate together disparate electronic health record data on the patient so we can pull this into our analysis, so we can start to think about the genetics and genomics of disease progression, subphenotypes, and drug response. Within the International IBD Genetics Consortium, we are very much interested in collating and sharing some phenotype data so that we can begin to undertake meta-analyses to identify regions of the genome associated with these phenotypes. So we have started to have some success with these drug response phenotypes. And so I'm showing you here a study that we published back in 2019. It was led by Alex Sazanovs in my group. So we measured anti-drug antibody titers, so the two anti-TNF therapies. So in this cohort, 532 patients had developed antibodies to anti-TNF. 708 patients hadn't developed antibodies to anti-TNF. We then undertook a genome-wide association study to identify loci associated with antibody development. And rather unsurprisingly, I guess, we found an association within the HLA region of the genome. And if we look at the risk of developing antibody titers, if you carry HLA DQA105, you can see that if you carry one copy of this is a dominant effect, which you can see here, the rate at which you develop antibodies against your anti-TNF drug is approximately doubled. And this allele is carried, it's a common allele, it's carried by about 40% of Europeans and about 20% of individuals in other parts of the world, such as Asia. So it's a common effect, and it more than doubles your rate at which you develop anti-drug antibodies. And developing anti-drug antibodies against anti-TNFs or any drug is bad news, because then that essentially clears the drug out of your system. And if you were previously responding to that drug, you can lose response. So now I want to go back to this problem of moving from maps to mechanisms, and I'm showing you here a region of the genome which is associated with IBD risk, and this is a kind of locus zoom plot of that region, and you can see that in this region there's a gene called integrin alpha 4. So the reason why this is relevant and interesting is because we know that integrins are expressed on the surface of lymphocytes. They bind to their receptors, and that facilitates the migration of lymphocytes through the endothelial cells and into extravascular tissues, such as the gut. So integrin alpha 4 forms a heterodimer, either with beta 7 or beta 1. The alpha 4 beta 7 integrin heterodimer, the receptor for it is expressed exclusively on the gut. And so it's via alpha 4 beta 7 that enables lymphocytes to migrate through the epithelial layers into the deeper extravascular tissues. So integrin alpha 4 is a really strong candidate for IBD risk. And indeed, we know that to be the case because vedolizumab is a drug which exists, which blocks alpha 4 beta 7 and is effective in IBD risk. But we wanted to try and set ourselves a challenge of how can we convince ourselves, if we didn't already know that integrin biology was important in IBD risk, how could we use the tools that we have available to us as geneticists and genomicists to conclusively prove this? So we obtained lots of EQTL data sets and started to co-localize, as many people do, our IBD GWAS hits with gene expression signals with EQTLs. And there was one data set from Ben Fairfax and Julian Knight and colleagues where we were able to have very robust, strong co-localization with our IBD GWAS signals. And what we've shown is across multiple different IBD loci that contain genes that are involved in integrin biology, yes, integrin alpha-4, but also ICAM1 and integrin alpha-L, we're able to co-localize EQTLs in stimulated monocytes with the EQTL for the integrin gene. I think that's a really important point to make, because I think what this shows is that triangulation was really relevant. We have multiple GWAS signals, all containing integrin genes, and actually across multiple of those loci, we're able to find consistent co-localization. And just to finish off the story, it was always the case that the IBD risk-increasing allele co-localized was driven by the allele which increased expression of the integrin gene. So if we hadn't already been onto the integrin, the importance of integrin biology in IBD, this would have put us onto it. So this was a great finding, and we thought, well, that's, it's comforting. We've managed to replicate some known biology, but how do we take this idea forward and try and use this approach to identify new drug targets? So what we started thinking about was how can we make the best EQTL maps for IBD? And so driven by that observation around these stimulated monocytes, we built and designed the MacroMap study. So what I'm showing you here, I don't know why it's animated like that. Anyway, hopefully now it'll be stationary while I describe this to you. So I want to describe the MacroMap study to you. So this is 200 iPSC lines, which we've differentiated into macrophages and basically stimulated the macrophages in a bunch of different ways and harvested RNA at different time points. So you can see we have some precursors and we have these naive monocytes where we've harvested RNA after 6 hours and 24 hours, and then we've taken those naive macrophages and we've stimulated them across 10 different stimuli and harvested RNA after 6 hours post-stimulation and 24 hours post-stimulation. This is an absolutely massive RNA sequencing experiment. It contains over 6,000 RNA sequencing libraries. I'm just going to take some time to give you a couple of vignettes from this work. So we're keen, obviously, to map EQTLs, and in particular, response EQTLs, and we used MASH-R to model the effect sizes across the various different stimulation states so we could settle on what was a true response EQTL. And what I'm showing you here on the x-axis is the number of EQTLs, and in the kind of pink color is the proportion, shows the proportion of those which were response EQTLs. So you can see after doing all of this, the response EQTLs are actually in the minority compared to the EQTL effects that are shared with baseline. So those of you who were here yesterday will have heard me ask Gosha the question about how many of these signals that you see after response are just driven by an increase in gene expression. So this is a question we looked at in our data, and basically to cut right to the chase, approximately half of those response EQTLs it looks like are actually bona fide changes in the beta coefficient of the relationship between genetic variance and gene expression, because there is no significant change in gene expression following stimulation. Okay. So while we're hearing now that there's a minority of EQTL effects which are response EQTLs, I just want to say that actually once we've done our co-localization with genome-wide association studies, and we took a very broad approach to this, taking not just IBD, but a host of different immune-mediated diseases, neuropsychiatric traits, basically what we could get our hands on via the GWAS catalog. Actually, those response EQTLs are very much overrepresented in what you get from the co-localization. So you can see here for CIL6, only about 28.4% of EQTLs are response EQTLs, but actually almost 50% of the co-localizations you detect are driven by those response EQTLs. So thinking then about how we can create the ultimate EQTL map for IBD, we embarked on a study using single-cell RNA sequencing, where we've ascertained, or we are ascertaining gut biopsies from 300 healthy individuals from the terminal ileum and the rectum, the two most common sites for inflammation in Crohn's disease and ulcerative colitis, and from 160 Crohn's disease patients. And the patients had to have active inflammation in the terminal ileum. And we also got a blood sample from the Crohn's disease patients as well. We then generate single-cell RNA sequencing, and this is a huge team effort, and I've just put some of the names and the faces of the people involved, with a view to then undertaking EQTL mapping. So I just want to show you here what we're getting from this kind of first phase of the data analysis. This is across 50 Crohn's disease cases and 72 non-IBD controls. We have almost 450,000 cells, and we've been able to identify 49 different cell types, which we've grouped into the 8 major populations that I'm showing here. And if we break them out, it's probably too small for you to read, but you can see here a kind of schematic of the various different cell populations. And what's really exciting is within this kind of epithelial cell cluster, we're able to assign epithelial cells to the crypt-villus axis. So epithelial cells that are at the base of the crypt, up to the middle of the crypt, and then actually the epithelial cells that are at the top of the crypt that are actually sensing the contents of the gut lumen. And very importantly, this population of mesenchymal cells, which has been recently shown to play an important role in IBD. So one of the things we were keen to do, because we've got a very large number here of cases and controls who've undergone single-cell RNA sequencing, was to undertake some differential gene expression analysis. And we do that differential gene expression analysis in the various different cell populations that we've identified. But because we've got a large number of cases and controls, we actually thought, well, we'll try and have a replication arm here. And so we split the cohort into these two groups, which I'm showing you here in green and blue, and then just asked what's the replicability of these differential gene expression signatures across those two groups. And actually, there was profound difference in the replicability of DGE across these different major cell type populations. So you can see in the top row, we have good correlations in the fold changes that we're picking up in our DGE experiments across stem cells, enterocytes, secretory cells, even B plasma cells. But actually, for some cell populations, the replication rates, the fold changes that we're seeing in our discovery and replication cohort really don't line up. And so why could this be? Well, if you look at the cell populations where the replication rate is poor, they are immune cells, which obviously are sensing the environment and responding to environment. They're also the cells which are affected by the drugs that IBD patients are on. So any small differences that we would have between the two cohorts in drug usage will, we think, be driving some of the differences we're seeing here. So I think these are important lessons to think about as we're thinking about scaling up our single-cell RNA sequencing experiments and wanting to undertake differential gene expression analysis. How can we make sure we do that in a way where we can control as much as possible for things like the drugs that different patients are on, to try and increase the replicability of the findings that we make? So the next thing we did is, because obviously my group is very much interested in genetics, we're excited by the causal anchor that genetics provides to biology. We wanted to test whether these differentially expressed genes that we'd identified were enriched with heritability, to try and identify cell types that might be causally driving IBD. So perhaps rather unsurprisingly, when we did this with our differentially expressed genes, we didn't find any enrichment of heritability, likely driven by the fact that we don't have many replicating robust differentially expressed genes across the cohort, thus limiting our power to detect heritability enrichment. One thing we can obviously do is use our marker genes, so the genes that we've used to define the cell types that from our single-cell sequencing, and we can test those marker genes for enrichment of heritability in Crohn's and UC. And actually, you can see here that we were much more successful. The cell types with the little asterisks there showed significant enrichment of heritability, many different T-cell populations and myeloid populations. And just as controls, we also tested for heritability enrichments of these marker genes with height and education GWAS, and didn't find any enrichment, which is what we're expecting. Just a little point here that in disease, in Crohn's and UC, what you see is massive and very profound cell population changes in the gut. So, for example, myeloid cells, you see very, very few myeloid cells in the gut of healthy individuals, but in IBD, the number increases more than tenfold. So when we're trying to do our differential gene expression analyses, we're really underpowered to pick up differentially expressed genes in myeloid cells. You don't see any heritability or enrichment in the myeloid cell populations here. If we look at the marker genes, then we do see heritability enrichment. And so actually, when we're thinking about analyzing our single-cell data, we're going to have to take different approaches that are based on where our power lies relative to the number and ratio of cells in our cases and controls. So where next? We've recently started a large study in the UK called IBD Response, which is particularly thinking about trying to identify and build these predictive models of drug response. So this is a cohort of 1,300 IBD patients who are about to embark on their first biologic therapy. And the study was essentially built around the microbiome. So across these 1,300 patients, we get measure the gut microbiome, the fecal metabolome, the plasma metabolome. We have very detailed food frequency questionnaires and follow the outcome of the biologic for those patients. And we've also generated human genome data across the cohort. Samples are taking pre-treatment, again at week 14, and again at week 54. As I've said, the idea is around predictive modeling, but also building up a biorepository of samples, including organoids, because we expect about half of these patients to undergo colonoscopy during the course of the study. And we at Sanger have kind of bolted on some single-cell RNA sequencing to this. So we're generating single-cell RNA sequencing on the baseline samples, and all of the baseline samples, and all of the week 14 samples. So we can measure the transcriptome in the blood, but then also from the gut biopsies that we'll get from approximately half of the patients at some point during the disease course. And so you'll hear when John speaks that it's this type of longitudinal study design with very broad multi-omic profiling built around not only predicting disease response, but also using, for example, the pre-treatment samples to get at disease biology and understand kind of causal pathways involved in disease that we're keen to build on in DiseaseMap. Okay, so I'm going to just stop there and thank, firstly, all the patients who've donated and samples to the study, the folks in my team and our collaborators and the funders. And just to say, we are hiring for lots of our single-cell RNA sequencing studies. So if you're keen to come and join us as we try to move from maps to mechanisms to medicines, then please drop me an email. Thank you.
Thank you, Carl. In the interest of time, we'll turn the floor over to John Rioux, who also can't join us for some family reasons. But Dr. John Rioux is a full professor of medicine at the Université de Montréal and senior researcher at the Montreal Heart Institute. He is holder of the Canada Research Chair in Genetics and Genomic Medicine. He is a co-founder and leader in the IBD Genomic Medicine Consortium, the North African IBD, or North American IBD, and the International IBD Genetic Consortiums. John's research focuses on genetic studies to identify risk factors for IBD, functional studies to understand how these factors affect disease risk, and prospective multi-omic studies to identify biomarkers of importance for clinical outcomes. Dr. Rioux is the co-lead of the ICDA DiseaseMap working group. And I think we'll have a video from him now.
Unfortunately, you won't have a video. You'll have me in person.
Right, even better.
Anyway, so thanks so much for the invitation. And again, I'm sorry I can't be there. And would just like to underline the fantastic ICDA team that's been so understanding of me not being there in person. So in the ICDA reached out to a number of us a few years ago to develop disease-specific projects, how to go from disease maps to mechanisms, and we were all pretty happy with what we had come up with. And then they came back to us and said, well, forget about what you proposed. Can you come up with a proposal that actually brings all these different projects together? And so what I'm going to try and present to you today is a result of those discussions across what we call the flagship disease projects. And I'd just like to highlight the people on the screen here who had the very significant contributions in putting this all together. So when we think of complex traits, and I know all of you do, there are a number of common challenges and opportunities that we face. Obviously, you all know that hundreds of known loci have been identified for many complex traits, but that it's a bit of a challenge to go from locus to gene variant, and even greater chance to go to mechanism. And this is probably because we're lacking biologic context, as you heard in Anna's talk and in Carl's talk, where the genes are expressed and at what time is very important, and what networks these genes belong to. Very commonly, we've heard that most of the studies that have been presented to date lack diversity, and this at the level of study subjects as well as the researchers. And so this has a significant impact scientifically. So we need to address this. What Carl just mentioned a little earlier was highlighting the heterogeneity in disease. There's heterogeneity at the level of clinical outcomes, genetic loci, and overall biologic level, such that there, if we just lump patients together without taking into consideration this heterogeneity, I think we're missing out quite a bit. So this is an important thing that we're going to try and address. This audience doesn't need to be told that genomic technologies, and particularly the single cell technologies that have emerged over the last few years, have really dramatically improved our ability to look at the biologic context in healthy and in disease tissue. And it was highlighted by Anna earlier. Some disease tissues are just difficult to obtain, so many studies are limited to a single tissue. And in order to validate any of our genetic findings and mechanistic findings, we need models that are relevant to humans, not just the animal models that have been the workhorse for many years. So we sort of set out to, based on these sort of opportunities and challenges, what a ICDA DiseaseMap project would look like. So the sort of the challenge we were given was, can we create a consensus paradigm for going from maps to mechanisms at scale? And while potentially focusing on just a small number of diseases, can we sort of think of using this to build in the future? So the project obviously needed to have multiple diseases, multiple phenotypes. We didn't want to work in a vacuum, either within the ICDA framework or the broader scientific community. So we had to sort of explicitly map out those overlaps. A project should have a combination of primary tissues, cells, and generate cell lines from these same individuals. And importantly, any project that we propose should respect and promote the ICDA objectives of diversity and inclusiveness. And not a small component in such a project would be not just scientific discovery, but creating resources for the community that would propel other discoveries as well. And as you'll see, the focus of the project is really surrounding patients, the patient data, patient samples, and attempt to be transformative for the field. So the three diseases that sort of landed in the final proposal that I'm trying to present to you today are coronary artery disease, type 2 diabetes, and IBD. All of these have fantastic genetic maps, as you've heard earlier, although lacking in diversity. There are multiple ongoing efforts to achieve greater diversity. Another advantage of these diseases is they're fantastic international collaborative networks that we can build upon the existing infrastructure, and also already have connections outside of North America and Europe. These diseases also share a number of common risk factors, be they genes, be they pathways. And when you look at circulating biomarkers, there are a number of biomarkers that have been identified, be they inflammatory clinical markers such as CRP, cytokines, chemokines, and others. So we think that these are good diseases to study when building this approach. So what have we come up with as a proposed strategy? And looking forward to everyone's feedback. Is that going across these three diseases, there are a number of pillars that we're proposing to build. First is applying in a harmonized fashion a collection of patient-derived samples that will enable single-cell technologies. Obviously, the solid tissues are going to be different between the different diseases as different organs are affected. But to combine this with the body map efforts that will allow us to go across a larger number of tissues and tissues that aren't necessarily easily accessible, this will sort of form the case control comparisons as well as within case comparisons. Very importantly, in these same individuals, as much as possible, we want to be able to collect blood so that we can not only look at the immune cells, look at known clinical biomarkers, but as well go across number of different omic technologies, single cell, as well as proteomics, metabolomics, such that in the same individuals we can compare what's going on in tissues as well as what's going on in the blood, and to be able to develop models and bank cell lines for others to use that will allow us to validate and explore the disease mechanisms again within the same individuals. So the idea is that together we'll be not only collecting samples, but also generating data sets for discovery and sharing those data sets and samples where possible within the community as well. And we believe that in doing so, we'll be able to place genes within loci within biologic context, providing us with the power to identify the causal genes and pathways. Looking across diseases allow us to sort of tackle pleiotropy directly, and taking a look at population differences across all these. Highlight here that we want to also be able to do capacity building. That is to say that in countries where they're just beginning to do IBD research or type 2 diabetes research or CAD research, can we help in building infrastructure? And with the broader community, we'd like to be able to share biological resources and data resources, including validated models and assays. So can't go into the detail of every single aspect of the project, but thought that I'd show you just a few highlights. And so, for example, in the inflammatory bowel disease space, that Carl just presented to you. We are proposing three themes. The first one is being able to look at the disease assessment. And here this is really trying to get at disease heterogeneity. So taking individuals that have just recently been diagnosed and following them longitudinally with sample collections so that we can not only see what their baseline status is, but how the disease evolves in these individuals, and including component with disease flares. So trying to capture at home what's going on in the biology of individuals that are having flares of their disease, and what it looks like post-flare. Second is sort of building on what Carl presented just a few slides ago, which was, can we build upon existing infrastructure to go much broader in terms of populations and diversity for building a unique resource to look at treatment response. And the third proposed theme is to really focus on countries, as I mentioned, that either have increasing incidence or just less research infrastructure, and helping build infrastructure, be that physical infrastructure or personnel infrastructure in terms of training, and having a broadly interactive group of individuals and training opportunities. So in a sort of a graphical view, what we're thinking are the three themes of being able to sample longitudinally recently diagnosed cases, and look at disease treatment in a separate cohort. But if people are beginning therapies in this first, we will then obviously include them in the second study. And in common to all these, in the capacity building and the treatment response and the disease assessment proposals, we would all have a baseline sample that would allow us to look at disease heterogeneity. And we feel that flares and treatment are things that sort of trigger responses that we can use to better understand the pathways that are ongoing in these patients. And as Carl mentioned, there are ongoing studies in the UK, and we've just completed a pilot study of the first 175 and aim to get 575 patients in Quebec, where we do broad multi-omic screening at baseline, that is pre-treatment and at early assessment, and following them for 1 year. Similar to what Carl presented, we would like to sort of expand on this in terms of diversity and enlarge the sample size. Switching to coronary artery disease, and highlighting the group that had a major important contribution in developing, and not just the field, but this proposal. As I mentioned earlier, trying to go across tissues, blood, and models. Why CAD? As this wasn't presented earlier, Carl and Anna did such a great job of presenting why IBD and type 2 diabetes. As most coronary artery disease is one of the leading causes of human death. And although that lifestyle changes and pharmacologic sort of interventions have had a major impact on disease, there still remains a very important impact of CAD, not just in European-derived populations, but the positive effect of these approaches have not been translated across all diverse populations. And sort of a particular opportunity for CAD is that most treatments do not target vascular wall biology, and it's because there's not much, although there's a lot known, there's not systematic in large cohorts. So what we'll be presenting for CAD is a real focus on the vascular disease biology. So the component for the CAD, highlighting the fact that the project really aims at getting a better understanding of vascular wall biology. And here what we're talking about, is at different stages of disease in particular. So being able to develop a single cell atlas in patient material, and you can imagine how this could be used, as in previous talks, in terms of better understanding regulatory networks, defining EQTLs, and really putting an emphasis on drug development in this area. So the idea here is just some numbers in the U.S. There are about 3,000 heart transplants in the U.S. per year. And coronary artery tissue is already being harvested at multiple institutions, but this is not being done in a coordinated or harmonized fashion. So the idea is to build upon this and develop a federated collection of flash frozen samples, not limited to the U.S., but a number of would want to collect throughout a number of different sites internationally, so that we can have achieve 1,000 transplant recipient coronary tissues. And as mentioned for IBD, not just tissue, but also blood, so that we can go across the solid tissue and blood. And here's a very important connection with body map, because the body map that was presented earlier will provide the opportunity to look at different vascular beds across the individuals collected, and you will expect that there's going to be a range of arterial disease in these individuals. So a nice spread in combining these two will provide a very unique cohort. So what's being proposed here again is to be able to look at disease progression of atherosclerosis, to really enable a better interpretation of GWAS, to get a better understanding of cellular trajectories, not just for the endothelial cells, but also for the hematopoietic cells. And this is building upon a very important infrastructure that's already been put together by the investigators, highlighting it even more here. There's been a tremendous amount of effort put into the Impact of Variation and Gene Function Consortium. Here's Jesse Ingritz and Tom Katermos, Million Veterans Program, Tim, and for example, Dan Rader for the UPenn Biobank. So bring all these resources, the infrastructure will really help jumpstart this component of the project. And to highlight on just one particular aspect of type 2 diabetes, as you heard earlier, type 2 diabetes has a huge impact worldwide, but there's lack of diversity, as Anna presented, and the existing resources are somewhat sparse in terms of beta cells, definitely sparse in terms of diversity, and therefore that impacts on what data sets are actually available currently to be able to go from GWAS loci to genes and mechanisms. So, as Anna mentioned earlier, one of the key areas of focus for the type 2 diabetes is really developing this GWAS in a dish approach across a large number of individuals, across a diverse set of individuals, and across a diverse set of tissues and or IPS-derived tissues. And to be able to do that, I think there's going to be an important collaboration between the CellMap project, which is not disease project, and the type 2 diabetes group, the IBD group, and the CAD group to be able to build the appropriate models and to build the appropriate assays to go along with these models. That will not only help the current set of known loci, but loci going forward. So a very complementary approach to what's being proposed in CellMap. So the advantage also is to be able to combine donors that are from the population, in addition to donors who are from different stages of diabetic disease, much like in the CAD. So really getting a spectrum, because we believe that this spectrum is going to help us sort of better understand disease processes. And as for the other disease areas, type 2 diabetes has a significant infrastructure on which to build. So as I mentioned, in addition to solid tissues, the idea for collecting is to collect blood across from all these individuals so that we can not only look within disease, but across disease. And this is important as there's multiple different indications that are inflammatory processes that are in common across IBD, CAD, and type 2 diabetes. And having the same individuals represented in both the solid tissues and in the blood samples will allow us to benchmark what biomarkers we identify in the blood with what is found in the solid tissues. And to be able to validate mechanisms both within this project and within the community, we aim to develop a number of different cell models, adipocytes, endothelial cells, epithelial cells, monocyte macrophages, hepatocyte-like cells, mesenchymal stem cells derived from the same patients as above, and to really work on harmonizing protocols and assays along with the CellMap project. So really just touched upon, little vignettes, but hopefully this doesn't give you the impression that these are, sort of projects going off in their own direction. Just want to highlight some interesting aspects across the different projects. But here, truly, the idea is to collect across these diseases, solid tissues, blood, develop models, really to do so in a fashion that both the data generated from single cell analyses, as well as biological samples that will enable others to do single cell analyses or bulk analyses as technologies evolve, really so that we can advance our ability to interpret GWAS studies and go from causal loci to disease mechanisms. So just like to thank in particular the people that are on the screen, including all the flagship disease member project members and the ICDA leadership and staff who've been extremely supportive, and any mistakes, silly things that you heard me say, don't blame them, blame me. Thank you for your attention.
First, I'd like to thank John. Delivering a talk that starts at 5:30 in the morning is always a difficult, if not ambitious thing, and I think you performed admirably considering the context and the time zone differences. I'd be happy to open the floor for a few questions. There will be a panel discussion at 2 that focuses primarily on the DiseaseMap efforts that Chris Whelan will moderate a bit later. But let's, as it's fresh in our minds now, maybe just take a few minutes out of our lunch break to see if there are any kind of key questions that people would like to address now. Yes. Oh, yeah, go ahead.
Thank you. Actually, my question is for Carl. I saw in your last slide that for future studies, you are planning on doing the transcriptome on the blood cells. And I'm wondering if you are considering deriving macrophages from monocytes isolated from the blood cell and comparing cases and controls and also the macrophages that are derived with the ones isolated from the intestine tissues and see whether there is a correlation, because there could be room there for escalation, for scaling this type of studies if those type of cells were a good proxy for what's going on in the gut.
Yeah, so really good question. So thinking about that at a very high level, one of the reasons why we are taking gut biopsies and blood samples is because we are very keen to compare the immune cells that are in the circulating blood versus the immune cells which are tissue resident. And so one of the things we will be doing as part of our current study and in IBD response and moving forward are those comparisons. We are also building biorepositories that would enable us at a future date, if we wanted to do exactly as you describe and derive macrophages from blood cells or from the gut biopsies. And we're also, it's not the immune cells or macrophages, but we're also making organoids from the gut biopsies we receive, with the idea of that then allowing us to do downstream functional experiments, perhaps even with the gut organoids, let's say, by perturbing the microbiome and looking to see how the cells in the organoid respond to that particular perturbation. So yes, we very much have the experiments that you describe, the type of experiments you're describing in mind, but at this present time don't have any plans to, for example, derive macrophages.
Thank you.
Any other questions? We have a few on Slido that we'll address, I think, in the panel for a little bit more of a plenary view. But any other burning questions that people have here? All right. Well, let's thank our speakers again. And thank you, John, for joining virtually. We'll see you a little bit later today, hopefully after you've had a bit more coffee. And we'll break for lunch. As a reminder, for the partners' lunch, you should know if you're a member of that. That'll be up on the 4th floor. For the rest of us, it'll be in the restaurant where breakfast is served, or where the reception was, right? Storm?
Yeah, where breakfast and dinner was served yesterday.
Where breakfast was served.
Same place.
And we'll be back at 1. This last session, we're talking about some very exciting things, and it's my pleasure to open the last session with Keynote and introducing our next keynote speaker, Oliver Stegle, who obtained his PhD in physics from Cambridge and did his postdoc at the Max Planck Institute for Intelligent Systems in Germany. He was a group leader at EMBL, the European Bioinformatics Institute, before being appointed a full professorship at Heidelberg University in 2020. He is the head of the Computational Genomics and System Genetics Division at the German Cancer Research Center. His group focuses on computational methods to unravel the genotype-phenotype map on a genome-wide scale, working at the interface of statistical inference, machine learning, and computational biology to integrate large and heterogeneous data sets at the single-cell level. And Oliver will be joining us virtually, so we'll have another virtual talk. Take it away, Oliver.
Apology for not being here in person. Unfortunately, due to sick kids, I couldn't make it. But I still hope I have something to share that is of interest for the discussion we've heard over the last 2 days. I will present on really advancing the tool chain for single-cell genetics, both primarily from a computational perspective, but also some experimental design that hopefully can feed into the discussions here that we're having. we want a recap on why we want to carry out genetic analysis at single-cell resolution. We heard several approaches and success stories and future plans to conduct such designs. But I think that the key point, right, is understanding this path from variant to function by being really cell-type specific to understanding how do variants propagate functionally, both in cis, but then ultimately also the level of trans downstream in cellular programs and cellular networks in a cell-type specific manner. But I think it's also important to realize that we really don't want to stop here at cell types, but we really want to use and leverage single-cell data sets and single-cell technologies to go in and are able to map out the trajectories of cells as they, for example, progress from a healthy cell state in a disease cell state, or how they progress across aging or other dimension trajectories. It can be both developmental within sample, but they can also be across people, understanding these cell states. And for the cell state mapping, here's a figure that I borrowed from the Lifetime paper in setting out this sort of broad vision. They really add value in being able to conduct this at the single cell level and then being able to map germline risk factors along these trajectories and cell state changes. So what does it need to make single cell genetics work in a reality? One is experimental advances. I want to be brief here, but I think it's just remarkable to remind ourselves within a decade, we went from the first proof of concept to commercial technology like 10x genomics and others that allow us to assay hundreds of thousands of cells in each experiment and bring the scale we require. We've also now multi-omics technologies, being able to assay variation in single cells in parallel at multiple omics layers, which allows us to look at unprecedented resolution and connectivity, something I will very briefly at the end hint at, primarily attack sequencing and how we integrate with RNA sequencing. But what I want to place my focus on today is the analytics. What are the questions and challenges of integrating these data sets? We have yesterday heard beautiful talk about MERFISH and other spatial in situ sequencing-based assays that allow us to now map out tissues in at spatial resolution, even subcellular resolution. And it's just one example of the type of things that I think we should really investigate and require to make genetics at single-cell resolution work. This is joint work with Omar Baratka's lab, where we set out to really tie together single-cell sequencing-based information from dissociated cells, single-cell RNA-seq, for example, with extremely high resolution at cell types and cell states, and then mapping these variations onto the spatial coordinate system that is, for example, being provided by Visium or other spatial transcriptomics technologies that may or not offer subcellular resolution, but that definitely offer the ability to understand tissue histology and understand how variation from cell states is actually manifesting in tissues. And excitingly, these types of approaches are even applicable if you don't have population scale variation on the spatial omics side, but we can use them as a reference coordinate framework to map variability that we see now from single-cell sequencing in many of these examples we heard about over the last 2 days. Another example that I want to dwell on a bit deeper, because it really gets to the meat of this presentation, is the questions about how we combine population scale sequencing from hundreds of people and possibly combined also multi-omics measurements, so we either assay one or single omics layers from the same individuals, and think about how do we make sense of these types of data sets that are now emerging and that are coming together from a computation perspective. One point that I think needs to be made is that infrastructures to handle the data that also was mentioned by several speakers are really critical. Here's an example of some of our work developing Python ecosystems to map out variation at RNA attack level across samples. There's an enormous logistics of thinking about which samples belong to which individuals, which assays are tied up, and maybe even which cells are profiled at multiple omics layers. And there's community efforts to do so, and many of these tools are already able and capable of going towards genetic analysis and the scale that we're talking about here. The second question is how to make sense of multi-omics data sets and data that comes from these types of multimodal, multi-individual profiling efforts. And I would like just to sort of draw attention to this one way how to represent these types of variation. It's essentially a matrix where we have different omics views or different omics layers that might be different assays we are applying to the same set of samples. Let this be RNA accessibility, methylation, or other dimensions. And then in columns, different samples. And these samples, again, cluster in groups. For example, group 1 might be one individual, group 2 a second individual, and so forth. So we need to account for this grouping structure, both in the molecular dimensions we're looking at and in the dimensions of individuals that we're assaying, and respecting, accounting, and modeling for these dependencies within these types of data sets we're now creating is obviously going to be really important to maximize the value of projects of the scale that are envisioned. And here's maybe just a representation, bird's eye view But what are the tools that community has been developed? This is mainly really driven by efforts like the Human Cell Atlas and a large computational community that thought about, how do we tie together data sets? And there's methods for horizontal integration where we really think about we measure the same thing, the same single cell RNA-seq in different people. And so the anchor that we're actually using on experiments is the molecular features, the same set of genes that we are assaying at multiple different independent experiments. Well, what I think in the genetic context and efforts like ICDA will be more the prevalent integration scheme is something that we denoted vertical integration in this review, where we embrace the idea that we measure these different omics modalities, for example, methylation, accessibility, transcriptomics, or others, in the same set of samples or the same set of cells, or maybe both. And this enables and brings fantastic opportunities for integrating and leveraging genetics or inter-individual variation as an anchor. And I think in this community, I don't have to convince you that this is a useful idea and really goes along the lines of many of the core principles of the projects we're discussing here in this fantastic conference. So just want to briefly talk to you about some of the tools we developed for this type of integration questions, and then also outline how we apply them to really make single-cell genetics work in practice. So for us, as an example, I want to just walk you through an approach we devised a few years ago with Wolfgang Huber and other collaborators, which is we term multi-omics fact analysis, which essentially sets out to make sense of these sort of connected multi-omic matrices of different cells and or samples being assayed at multiple levels. Here, 3 methylation RNA accessibility is an example. And these ideas, they're fundamentally statistically, in a way, simplistic. And that also turns a lot about their beauty, because all we're doing is actually generalizing principal component analysis or fact analysis to this sort of multi-group design. So we're still decomposing this big matrix into smaller matrices, very along the lines of what everybody's used to the PCA. But the core idea is that one of these 2 matrices is now shared across all these 3 factorization problems, while the loadings, which maps how do these factors, these components, map onto these individual features, they're specific to modalities. But there's a joint manifold that represents what's the variation across individual cells, and that obviously ties everything together and allows us to reason about joint cell states and variability in this new coordinate system that we're ultimately interested in. So there's a number of challenges, and I think one that I think was very clearly also highlighted in the presentations over the course of the last 2 days is, to bring this dimensionality reduction idea that we're all familiar with and use daily in genetics and genomics together with spatial and temporal modeling. And that's something we've actually done earlier this year. Britta Welton has put together, I think, a really nice principled approach that allows us to really now think about, data cubes. We have data sets with different omics layers here in the Z stack. We might have different groupings, which might be, data from 1 or hundreds or thousands of people. And we do measure increasingly different omics modalities across space and time. And we want to be able to leverage and make use of these spatial temporal covariates. Then, for example, breaking down variation still to components. These components might capture shared variation that's really uniquely observed across all sample groups. Groups might differ, for example, different disease groups, case control settings, or there might be stochastic variation that's not even linked to temporal or spatial profiling. And this is a very generic toolkit that I think we can deploy to a number of questions. We've used it, for example, here in longitudinal single-cell multi-omics profiling, where we single-cell data at multiple omics layers, and we identify developmental lineages using these approaches. But they're equally applicable. I think that's really exciting to, for example, longitudinal microbiome profiling. We have different groups, different sample sets, and we can really understand what's the temporal dynamics of disease trajectories going forward. And many of the ideas I present further actually use Mephisto and Murpho under the hood. And I will reference to this in due course. All right, and with this, I would really like to sort of think about, switching over to this genetics questions, which is to think about how we can now use these types of omics not only to understand variation on the level of samples or individuals, but also tying it to genetic factors, understanding how variation in our genome, individual variants or polygenic risk scores, links to expression changes in genes, but doing this also in a way that respects different cell types and cell states. I motivated this earlier. We want to really mine the single cell data and obviously dissect it to fullest extent to then understand these dependencies. And to do so, I want to walk you through a data set we created with collaborators in the UK a few years ago, because I think it still highlights a number of sort of principal questions. And I hope I can also provide some insights on how to design some of these experiments going forward. We looked at data from the UK Human-Induced Protein Extension Initiative, which is a joint effort with the collaborators across the UK, and it has established a panel, a population scale panel of normal reference human iPSCs drawn from healthy volunteers in the Cambridge area in this specific case. And while we learned a lot about IPS biology itself, I really want to just focus on one use case of this resource, namely to take these iPSCs and to push them using cellular differentiation towards disease-relevant cell types and lineages. And we've done so in endodermal lineages, which I'm not going to present here, but we've also done so in the context of neuronal lineages, which I want to talk here to you about, and then also discuss with you how we actually mine value and think about this data from a computational perspective. So this is data that was generated in Daniel Gärtner's lab at Sanger in close collaboration with Florian Murphy's lab. And what we've been doing in this study is take 215 iPS cell lines drawn from this Hipsky reference population of iPSCs. We differentiated them towards a neuronal fate, and we did so in a pooled manner. We actually looked at pools of roughly 20 individuals. So this is really, the first step towards representing population variation in a dish. And these pools, in this case 10 pools, were single cell assayed at 11 days, 30 days, and 52 days towards differentiation in this neuronal fate. We've also considered a stress condition. So in addition to the naive state, there was also a Rotland stress applied, which is an oxidative stress that we can obviously utilize and exploit the fact that this is a cellular differentiation system that enables such perturbations. Now, you were wondering about pooling. I just want to sort of remind ourselves that this is now, I think, a fairly robust approach to multiplex molecular biology at scale. Essentially, these pooled populations, after single cell profiling, every cell has to be originating from one of the individuals in the pool. And there's now very robust computational workflows to demultiplex these individual populations based on the genetic barcode they consist. So very briefly, essentially what we're doing is we're assaying germline variants we can read off from the single cell RNA sequencing, and then we can map each cell to the donor of origin. By the way, I should mention, this is not only limited to decomposing individuals, but can actually also give rise to subclonal structure. We've actually demonstrated in the context of skin cells, but I think also clonal hematopoiesis might be tractable and even get variation within individuals using such deconvolution methods. Now, once you've done that, we essentially get these global maps of variation in the context of this neuronal differentiation system. Day 11, these are mainly progenitor cells. Day 30, you see this neuronal population here in green emerging dopaminergic neuron and serotonergic neurons. These change cell state and quite substantially approach maturation towards 50 days of differentiation. We later on will want to ask the question whether actually the regulatory landscape between these two populations changes as well or remains identical. And then here on the right, you see the changes upon the stress response. So the first question I quickly want to walk you through is asking, do these differentiation changes occur in a universal manner across a population? And the answer is unsurprisingly no. There are substantial differences if you take such a collection of iPSCs and differentiate them. If you, for example, measure differentiation efficiency defined as what's the fraction of neurons you get at the end point of this differentiation assays, you can see there are lines that basically make almost no neurons, only 10% or less of the cells of neuronal origin at the end, versus other cell lines have an extreme high efficiency, 80–90%, creating neurons. This is actually robust across replicates that we are observing here. So you wonder, is pooling the reason for that? And I think that's also thinking about many of the design choices in the experiments we saw in the context of ICDA. So I put that slide in here to just make the point, actually pooling in our hands in this specific system doesn't explain this variation. If you differentiate lines one by one versus pooled, you get very little differences. This is really a cell intrinsic property of a specific iPS cell line, whether it is going to make neurons or it is not as efficient in creating these specific cell types of interest. What is, however, an interesting effect of pooling you need to think about when scaling these experiments is that upon differentiation, the representation of donors, or every line here is every color here represents one cell line. The representation of different individual changes, and obviously you're not ending up with a balanced design at the endpoints. And that is the price to pay for multiplexing molecular biology. And you need to think about how do we take this forward in analysis. Actually, we set out to create specific analysis approaches based on linear mixed models that account for the fact that if you do pseudo-bulking in these populations, here, for example, serotonergic neurons in light green and dopaminergic neurons in dark green, you basically create pseudo-bulks of varying extents of certainty. You may a lot more about some individuals than about others. if you haven't added as many cells. And that's something we need to really think about the analysis. And there's also a paper I refer to you that discusses this in greater detail, how we can propagate this uncertainty in our genetic analysis, and we can really map cell-type specific EQTLs. Here's, for example, an example of an EQTL that's specific to certain neurons and is not visible at dopaminergic neurons. So just a few words about the obvious question, how many EQTLs do we find? It's thousands. We have power to detect between 1,000 and 1,500 genes with at least one EQTL at a relatively stringent FDR. There are power differences by cell types, which are explained that we have many more cells observed in these progenitor cells than in its later stages. But I see there that there's substantial replication and identification of neuronal and neuron-specific EQTL signals in this cellular system. We also wondered, how do these maps compare to what we have established as a community in primary tissues? If we think about sharing of EQTLs with brain GTEx, we do see that upon differentiation, these EQTL maps start to resemble more and more, also after maturation, what we do see in vivo. So this gives evidence that these cellular models do recapitulate what we're interested in. However, it is important to realize this is somewhat an apples with bananas comparison. We're comparing cell type resolution EQTL maps in a cellular model with bulk region level brain area EQTL maps as we have them at the moment available in resources such as GTEx. Nevertheless, there's still questions. Are these designs powered? Can we actually do single cell EQTLs at scale? And I think if you aggregate this, if you count for the fact that our data is multiple cell types, and what you really want is actually make comparable data that is sort of equivalent to pseudobulking, you can see that sample size adjusted, you actually get very similar power from single cell EQTL studies already to date. And this is just one example. There are many other studies now have shown that really single cell genetics is now feasible at scale using today's technology, and it will become even more tractable going forward and maybe be a viable alternative to many of the bulk assays we're conducting. So, just a few comments on disease. We can identify co-localizations. Here's an example of a GWAS schizophrenia hit for which we find a co-localization signal in some of our EQTL maps. We can also think about globally. And I mean, the whole point I want to make is that these cellular models do replicate to some significant extent what is visible in vivo, but there's also additional new signals that come about when using such a process. And I think these are lessons learned we can also take forward when thinking about the next iterations of experiments and studies for our community. I would like to now in the last few minutes, really switch back to the analysis. And I think this is some principal approaches that might be really relevant when thinking about how to mine these data and get the most out of them. And I started my talk by telling you that we can take single cell assays and understand what are the principal molecular variations, differentiation trajectories, cell cycle. We can decompose the different coordinates across cells differ. And the question is, how can we leverage that information when doing genetic maps? We don't want to discretize cell types. We don't want to pseudobulk cells into low, medium, high across a differentiation trajectory like I have done now. Essentially, I've only shown you discrete cell types based on marker genes that we can deduce post hoc, basically using transcriptomic signatures. What you would like to do is map genetic effects in single cells. And that's obviously a challenge because one and the same cell you only observe from one genotype. So you can't directly compare it to its virtual sister cell that originates from another donor. But what you can do is you can clump together cells that are transcriptionally related in a smooth and soft and continuous manner, and thereby in little mosaics and little neighborhoods compare the impact of genetic variation on fine-grained transcriptional differences as you observe in them. And that's an idea that Anna Kumo, together with Tobias Heine, have putting forward in the lab, and really generalizes the idea that we want to basically not estimate genetic effects in discrete groups, but we want to get effect size estimates in each individual cell. And the trick we're using is essentially using these transcriptional neighborhoods that we can identify from the global transcriptome itself. And that allows you to fit an over-parameterized model where we have one effect size for every single cell, which is as many parameters as data points, but we can tie them softly together and reduce degrees of freedom to make these approaches tractable, calibrated, and even scalable to large data sets as we're now collecting them. Here's just a few examples of how that looks like. If we think about this dopamine neuron populations that I talked about earlier, this is basically now looking at one cell type in our previous study, this dark green population here. And we're asking, is there substructure within this cell type that manifests in differential gene regulation? And the answer is yes. it's roughly 10% of the EQTLs show the substructure, so quite a significant and appreciable amount. And what you're seeing on the right are just some examples. This is an EQTL at Win3, TBCA, and what is shown here is not the expression level of those genes, that's important, but it's really the leading effect estimate. So the effect size estimate of our approach for these individual cells by integrating in some smart way across the cellular neighborhood. And you can really see that there are sort of different grouping structures. Those look very similar. These look very similar. We can even cluster those EQTLs by asking, where do they manifest? What are the cell states where they manifest? And I'm showing you this example because I think it's a really important way to think about whether we shouldn't use genetics and gene regulation to define what our cell types. So rather than clustering our UMaps based on the transcriptome, I would argue we should consider clustering them based on these genetic effect profiles and then using those for even defining cell types, because these are functional subcell types that actually manifest in differences in gene regulation. Actually, you can show that if you just compare our approach to de novo subclustering of cell types with more and more subclusters, you actually never reach as much power as your approach using the method I described, that smoothly integrates and avoids the need to discretizing your data a priori, which is something we really should not do when we really want to maximize the insights gained from such single cell efforts. Finally, just one example, to think about disease subsets and disease fine mapping. We haven't really looked at this very deeply at this point, but here's just one example of a GWAS hit for sleeplessness insomnia that co-localizes with this gene SLC35E2. And what you can see that if you look at the right cellular subset that's identified as the most relevant one, you also increase power for co-localization compared if you use a much broader or more cell type agnostic approach. So subsetting your cells, subsetting the evidence to the most relevant cell states may actually increase power rather than decreasing power, because obviously you're reducing the cells you're looking at. So that's the point I would like just to make here in this example. And with this, I'm already coming to the end of my brief presentation. I want to maybe take you a little bit on an outlook. What do I think should we consider when planning the next steps, when we're planning efforts in ICDA? Everything I talked about and what we also heard about in other discussions over the course of the last 2 days was CIS, variant to downstream targets, immediate consequences. What we now really need to think about, what are the trans effects, what are the cellular programs affected, and there are a number of ICDA programs that have this vision and aim to get towards trans effects downstreams. Here are just 3 ideas that I would leave you with. One is multi-ohm data. I think particularly multi-ohm RNA attack provides fantastic opportunities to combine dimensional reduction with gene-related network inference. So we have preliminary results that are very promising, but to extend these sort of matrix decomposition methods like Mephisto and MOFA I talked to you about, by incorporating rich prior annotations about motifs, binding sites, and peaks, but also important signatures like covariation of transcription factor at the RNA level with variation in peaks where motifs correspondingly are expressed and downstream genes. And you can use these types of approaches to estimate where transcription factor is expressed, but you can also use these approaches to identify candidate cell-specific gene-related networks, which I think are really important to consider and should be really included in many of the efforts that go forward. The second idea that I think is also touching on many of the themes we heard about in this meeting is CRISPR screens and perturbation screens. This is work with Leopard's lab, where we've actually combined this population scale approach, so multiple individuals, multiple genetic backgrounds, with a genome scale CRISPR-I screen. And I just want to leave you with this idea that we now create matrices where for each cell, we know what is the perturbation, but we also have multiple genetic backgrounds. And this is really fantastic because we can use natural genetic variation side by side with engineered variation, integrate those. They're phenomenal, exciting statistical opportunities. We can use the variation that you see here between donors, the substantial variation from line to line that is actually overriding, if you wish, the signal that we see from the perturbations. And we can use that to leverage within actually causal inference methods to get much more robust estimates. Independent mechanisms principle is something we're using. And I think this will be a fantastic design to embrace and to think about to combine natural and perturbation scale approaches. And the final point is scale. And you will hear about this in Nicole Soranzo's pitch just after my presentation. Scaling single-cell genetics to tens of thousands of individuals will really be required to look at the downstream consequences of natural genetic variants, weak effects in rare, ultra-rare cell types, I'm very excited also thinking about how do these single cell approaches capture other variation in populations, environmental exposure, stimuli, life history, dramatic life events, and others. And I just want to leave you with one sort of other sort of sideline. In addition to my own science, I'm also now co-heading the EMBL Human Ecosystems Universal Theme, which really embraces the idea that we really need to get towards environment. And I think we should also think about the single cell data we're creating as a proxy for environmental exposures and stimuli. There was already indications in this direction from Gosha Trinker's talk yesterday. And I think these are interesting ideas to get an E, not only G, and tying this together. And I was very happy to discuss this further in the context of the EMBL Human Ecosystems themes. With this, I would like to thank the lab and fantastic collaborators. I think I acknowledged everybody along the slides, and I'm happy to take questions. Thank you.
All right, we'll have maybe open the floor for one quick, brief question. Go ahead, Cecilia.
Thank you, Oli. That was amazing, as always. So I really like the comment you made on the last slide, and we have representation from NIH in the room that have discussed with us the great, often overlooked importance of the environment in this type of study. And we discussed the exposome, which NIH is laying out a roadmap for, which we're excited about. I wonder if you could just talk to us in a cell context and cell experiment context, what do you think is the environment and how will you measure it? Is it various stimuli, or are you exposing the cells in various ways? If you can just talk us through sort of what you think we could do to glean insight in that way.
Yeah, it's a very good question. I mean, I think I have more ideas and maybe questions myself than answers at this stage. I do think we need a combination of approaches. We obviously need accurate ontologies for tracking environmental exposures in cohorts. That's a prerequisite, and the entire exposome community is embracing these ideas, and I think the standardization across cohorts will be very critical. However, I also think that molecular proxies of environments is something we should really seriously think about. We all know about the methylation signatures of smoking, the first sort of EVAs hits and others. That's a very, dramatic and drastic environmental exposure that you're exposing your body to. But there is really prospect that primarily, if you think about blood and immune cell states, there might be much more subtle life history events that we can now start to map out molecularly. And these molecular proxies for exposures will be much more robustly integratable across cohorts. We can measure them, they're quantitative. There's not only single cell RNA-seq, there's obviously also mutational signatures that come from more the oncology world. So I think these are interesting modalities we should think about collecting to create measurable molecular definitions of environmental exposures. And then you highlighted, already in your question, the mechanistic side. We need these stimulus types of approaches as well to create, primarily think about the mechanism and drilling down into what these things do. Once we have hypotheses, we need lab-based models, cellular models, and organoid models that we then stress with appropriate environmental exposures to then replicate these things are real.
Wonderful. Thank you so much. It's now my distinct pleasure to introduce our last speaker of the meeting, Professor Nicole Soranzo, who's head of the Genomics Research Center at Human Technopole in Milan. She's a senior group leader at the Wellcome Sanger Institute and became professor of human genetics in the School of Clinical Medicine at the University of Cambridge in 2015. Dr. Soranzo obtained her PhD in genetics at the University of Dundee and later trained at UCL. After a brief stint in pharma in the US, Nicole joined the Sanger Institute, where she started her own group in 2009. Her research focuses on identifying the genetic determinants of complex human traits, with a particular focus on blood and immunity. Nicole's also a member of the ICDA Executive Committee and DiseaseMap Working Group. Take it away, Nicole.
Thank you so much. And this is my slide. Yeah, so I just wanted to really thank Oliver for the introduction. I think it really sets the scene to what I'm going to try and tell you next. So this is really a project. In fact, the pitch is the continuation of a project that we thought now a few years ago, maybe 3 or 4 years ago, seeing how much of an impact the Human Cell Atlas and other single cell approaches have done in understanding and really giving us a refined, precise understanding of biological states within cells. And of course, I'm a human geneticist. Oh, I'm missing a slide. I'm a geneticist, so I've been engaged with the community in the last 15 years in discovering GWAS loci and trying progressively find map association and discover causative genes underpinning these associations. I've done a lot of work on blood and blood count differentials, among others, as intermediate phenotypes for immune and inflammatory diseases. And so there are some challenges we all know about the fact that much of the GWAS loci are non-coding. Many of them are uncharacterized, and indeed there's growing evidence that also rare associations tend to be harbored non-predominantly, but significantly within non-coding regions of the genome. So transcriptional activity is one of the components that can allow us to very rapidly read the function of the genes in individual tissues. And as mentioned many times before, there's still a huge challenge in understanding function, context, not only in terms of the type of cells or potential environmental perturbations, but indeed also how these genetic effects change in response, for instance, to biological processes such as aging and disease states. On the other hand, I also wanted to point the fact that, our research has really been facilitated by large-scale genomic resources. UK Biobank is one of them. And really, the exponential increase in knowledge that has been brought by the increasing ability to just go on a database and look for a variant of interest and quickly annotate a function. Function not only in a specific biological domain, but indeed, importantly, across many different domains of biology. In addition to the large-scale population cohorts, such as Biobank, we are also learning a lot from studies that focus on populations, such as, for instance, the East London Genes and Health, that are enriched for carriers of, for instance, loss of function or homozygous mutations. So we really thought, the timing is becoming mature, but we can begin to leverage ongoing efforts to recall volunteers and donors across worldwide cohorts, and to combine power single-cell transcriptomics with an increasing wealth of existing data, both molecular, genetic, but also importantly environmental, as mentioned before, and try and leverage this additional data layers to begin to understand the impact of genetic variation on biology and disease at the level of individual cells. And what's facilitating this thinking is the fact that the single-cell transcriptomics are becoming not only robust and cheaper, but progressively scalable to the level of populations. So really the vision for this pitch and for what we're trying to do is to bring the power of single-cell genomics to a population scale. And we want to do that by profiling activity of genes in not only hundreds or tens of thousands of people, but indeed tens of thousands of people. And so the idea is to add really a layer of information to this growing matrix that we're building. So there was a lot of discussion already in this meeting, very important discussion about the value of in vitro versus in vivo models, the value of using baseline information versus stimulated states, for instance. And of course, both are important and both give us information on different layers of biology. In the case of our cells of interest, peripheral blood mononuclear cells, these are cells that are readily available from tissues in vivo settings. They are, of course, proxies for function or can be informing function across a number of diseases. And interestingly, with a single experiment, we can capture tens, if not more, types of cells. So the project that we do bridge information that we get from genetic studies with also detailed exploration of function in ICDA. So thinking about, I just really want to focus on what we gain from doing tens of thousands rather than hundreds or thousands of samples. So really the idea of increasing this profiling at scale. Oliver has already talked about this a little bit. Just to remind you, first of all, is that there's a real opportunity to create a really definitive resources to characterize inter-individual variation in cell states in blood. And indeed, with the sample sizes, we're powered to not only discover the common cell types and the common genetic effects, but then we can go really quite deeply into rare cell types and rare genetic variants. And importantly, not only the cells and the cell states that we know already about, but also there's a huge potential for discovering new cell types and states. And Oliver was already mentioning the idea of coloring your UMAPs based on the genetic effects, so as a response to, as a function of what the genetic variant does in the population. Oliver has mentioned this ability to refine our understanding of genetic effects at the level of potential developmental trajectory, cell states, but also importantly in response to external perturbation. And of course, these models of QTL mapping as a continuous rather than discrete cellular process is going to, for sure, provide us with new information. Is mentioned already the ability to find map by, again, using this increased granularity of understanding of cell types. And importantly, underpinning all of this is the ability to move from a paradigm of QTLs that is only based from looking at the mean expression of a gene in a given cell to indeed a broader definition of transcriptional outputs. For instance, those considering the variance in expression of a gene rather than the differences in variance in expression of a gene rather than the differences just in means, or indeed allele-specific expression. And this can be done along trajectories of cells that can be defined in many different ways. Of course, going at population scales also allow you to really tap into the reservoirs of rare and private genetic variation. And of course, we're interested, particularly in the case of East London Genes and Health, but more in generally, the ability to quickly fish a gene out of a large gene pool and understand the transcriptional consequences of a large number of different cell types. Oliver has mentioned this really wish to define cell type defined cellular networks. And of course, you can think about a population, as a pool of cells, and you can think about genetic variation as effectively a genetic perturbation in a system. And so the ability to go very quickly into this type of data set and deduce how a genetic effect perturbs downstream regulation, it's really going to, I think, change also our understanding of dynamic systems. We talked a lot about these disease states, and of course, in a population scale, you can cut it in different ways, including by disease state. And so you can think about doing expression QTLs by drawing individuals from different disease categories from your pool of data. And this is just a very simple plot showing, for instance, if you take a resource like UK Biobank, how many cases you would discover if you had a sample size of 1,000 up to 60,000 people. And you can see that this brings into focus rare diseases as having sufficiently large sample sizes to be able to do disease aware EQTLs. And finally, there was a discussion about the exposomes and really the ability to tap, in this case, not at the cellular level, but indeed at the level of the entire system about genetic effects in response to different types of environmental perturbation. And not only as a measure of perturbation, but indeed begin to address causal inference in greater detail. So as I said, I think this is a complementary project to many of the ICDA proposals you've heard, because we're already all thinking about anchoring genetic variation to gene expression to function through genetics. But of course, there's a big component of projects that also look at PBMCs across the board. And so we can begin to think about how the information that we can draw from population scale informs also our understanding of these individual relationships. So when we thought about developing a single-cell project at scale, we thought, first of all, what are the operational considerations you need to deliver such a project? First of all, you need to have biological samples that are readily accessible in a predictable way. You need to have automated error-free processes, and that includes not only robust experimental pipelines, but also automated analytics and diagnostics that help you monitor in real time the quality of your data. So these were motivating factors for our pilot. I'm not sure how I'm doing for time. For our pilot, which we did in collaboration with Oliver Stegle, with Sarah Teichmann, Hilary Martin, with David Van Heel from Queen Mary's, and also with the colleagues from the UK Biobank. This was not a collaboration just of PI, it was indeed a huge team effort by many Sanger colleagues. And it's really important to emphasize how the scale-up of a project of this type really requires expert and technical knowledge, particularly in automation and data processing. And really a huge thank to all my colleagues that helped us in this journey. So we, as Ben showed this slide, we set off to ask, first of all, can we do reliably single-cell RNA profiling at scale in samples from deeply phenotype population cohorts? And what is the value of this data in understanding about characterizing comprehensively blood cell types, states, and also ratios in diverse human populations? We wanted to look at how we could generate and increase our understanding of genetic effects, both in cis and trans, in diverse populations. And finally, also to contribute to biomedical discoveries. I will say that the data is still being analyzed, so we don't have an answer yet to all of these questions, but we're working quite hard to get them. So we partnered up with 2 cohorts. One was UK Biobank. You all know about UK Biobank. It's a hugely valuable resource for the community. And in this case, we were able to access, through the sample application, blood from donors that underwent imaging in MRI imaging of 4 different sites in the UK. So we targeted for this pilot the analysis of 5,000 donors from UK Biobank. And, oh, sorry, I should have said, and apologies for missing this. This project was funded by Chan Zuckerberg Initiative, and really a huge thank to Jonah Kuhl that gave us really a lot of trust to develop the project. So very much, it's been a great collaboration so far. So as I said, and so in this case, in UK Biobank, we wanted to tackle 5,000 donors. We also, and indeed this was also prompted in part by CDI's wish to increase human diversity. We partnered up with another cohort, Genes and Health in London, with David Van Heel. And this is a slightly different setup in that this is a population that really focuses on volunteers from South Asian communities, particularly Bangladeshi and Pakistani origin, that live in London and in Bradford. And in this case, we have genetic data as well as phenotypes from health records, perhaps less of a deeper multi-omic phenotyping. But importantly, in this case, we were able to set up a community-based recall, and we were targeting to recall 1,500 people from this study. So the current design is shown here. Basically, every day, nurses collect fresh blood from volunteers at 5 different sites throughout the UK. The blood is picked up at 5 o'clock in the afternoon by a courier, and is shipped to the Sanger Institute and arrives by 9 o'clock the next morning. And so by 9 o'clock in the next morning, the sample is received, and then it undergoes effectively a whole day of processing through our pipeline that I'm going to show you in a little bit. And so it's really important that, of course, we have sound, robust, we're dealing with in excess of 70 samples per day, and we want robust processes to do that. So this is an experimental pipeline. So Stephen Watt, shown here on the left, together with a team of really good, talented technicians, set up a fully automated, hands-off pipeline to do the following 4 steps. We do immune cell purification using a fully automated robot system. We count the cells that come out of the PBMC preparation. We create balanced pools of cells by combining, informed by the cell counting readout. We rearray the cells and pull them and put them on a 10x Chromium machine. So all of this process is fully controlled through a LIMS system. A LIMS system that allows to make sure that we don't make mistakes, that we do these calculations efficiently and in real time. This is just a setup of the Hamilton robot that we use for this process, and indeed this is used for four different steps: for the PBMC isolation, for cell counting, for cherry picking and normalization, and also to create aliquots of PBMC that we bank and that will return to the cohort owners for further use. Of course, the experimental pipeline was hard to set up, but works incredibly well. It's very robust. In addition to this, we also needed to set up analysis workflows were equally robust. As you know, single-cell analysis is not an exact science. It's perhaps not as simple as just calling variants from a sequencing data set. So this required a lot of iterative processing and tweaking and a lot of thinking. And this is the current version of the pipeline. I'm sure will change again. What I want to say is that in order to accommodate this need to iteratively look at the data and process it and reprocess it, Vivek, here shown on the left, led a team of computational scientists, and particularly by Mathis Ozols, shown here on the left again, we were able to set up, again, a highly processive computational pipeline that does all the steps shown here, processes data very quickly, and can be basically not only manipulated to change parameters, but also indeed outputs automatically reports, including, for instance, cluster plots, cell type assignment plots, etc., quality on the spot. So we can go on a website and look for every sample that goes through our pipeline, what is the quality of the data. And this is publicly available. So just to go over the process, so we were able, as you see, the initial stages were stages of basically optimization. In about, I think it was in May of this year, we started scaling up production, and we are now reaching about 5,000 out of the 6,500 target donors collected and processed in our labs, with an average of about 400 donors per week. This is the plot of the QC pass cells that we retrieved. So we collected about 5.6 million cells to date, and there's a median of about 1,800 QC pass cells per donor that we can collect. So the data looks good, I think. So this is just to show that, for instance, we don't have significant batch effects. We, of course, have a lot of metadata and really actively looking at variation across all axes of variation, both technical, biological, to make sure that we can account for this information in the normalization steps. And finally, just a note, as I said, the value of going large is the ability to go quite deeply into the pools of cells. And so this is basically the number of the counts of cells that we get from the entire data set, so 5.6 million cells. As you see, we are using canonical approaches such as cell typist and Azimut. We can already call 30 different cell types. And indeed, even for the rare one, we have sufficient large number of cells pooling across different donors that we can begin to call EQTLs quite deeply in our cell pool. Okay, so thinking about the scale-up, what we'd like to do, well, first of all, just to remark, this is a fully portable experimental and computational pipeline. So anybody who wants to implement it could do that with, of course, our help, and could allow bench-to-bedside application, for instance. So what we'd like to do is to extend this current pilot to survey, ideally, perhaps in the order of 50,000 samples from multiple sources, of course, depending on funding, and both exploiting current capability, but also potentially new sites. And then we can, of course, think about extending this profiling in different dimensions, including using long-range technologies, for instance. So I think overall, the kind of possibilities, hopefully, that we can see is that a resource of this type could be landscape changing for the whole international community. So thinking about understanding the genetic controls on transcription, particularly in trans effects, which are underexplored, and cell type composition. In understanding better the transcriptional regulation in the context of genetic, phenotypic, and environmental variability. In understanding the molecular mechanisms that underpin disease risk variants in different diseases. in looking at transcriptional changes that are associated with rare genetic events, such as loss of function mutations, but also polygenic risk. In characterizing transcriptional signatures of biological processes, for instance, aging and diseases, in general application of epidemiology, environment, epistasis, and modifier disease, and of course, fostering the development of continuous development of analytical methods, as Oliver has already shown you comprehensively. And really, our aim here is to develop a resource that can be used widely by the scientific community, and hopefully that increases the resolution and scale at which we're doing human biology. So with this, I'd like to thank all of you and many, many collaborators that I listed before, and happy to join the panel for questions.